Hello and welcome to Lossimania. Today we're going to speak about the three important carboxylases. So the three important carboxylases are, first one is pyruvate carboxylase, second one is Propionyl-CoA carboxylase, and the third one is Acetyl-CoA carboxylase. We have a fourth one as well, but I'm not going to mention it right now. These are the ones that you have to know by heart: pyruvate, Propionyl-CoA, and Acetyl-CoA carboxylase. I'm going to go through them exactly where they are in the cell and their functions. So three carboxylases. These carboxylases are called ABC enzymes. Why? I'm going to tell you why. Because ATP is needed, biotin is needed, CO2 is needed, and it's ABC, as you can see there. And its function is to transfer CO2 groups with the help of biotin. So don't forget that its function is to transfer CO2 groups with the help of biotin. First one, pyruvate carboxylase. It's in the gluconeogenesis pathway. It converts pyruvate, which is a 3-carbon, to oxaloacetate to a 4-carbon. It is activated by acetyl-CoA, and it is in the mitochondria. It's activated by acetyl-CoA from beta oxidation. So it's activated by acetyl-CoA from beta oxidation of fatty acids. Propionyl-CoA carboxylase is the second carboxylase. Its main function is synthesis of succinyl-CoA from odd number of saturated fatty acids. It's a 3-step pathway, and it's when succinyl-CoA goes into the TCA cycle, it's a TCA cycle intermediate which gives us energy. So synthesis of succinyl-CoA from odd number of saturated fatty acids. That's the task of Propionyl-CoA carboxylase. Gives us energy. The continuation here, it's also produced from amino acid metabolism, and we have 3 amino acids. It's threonine, isoleucine, and valine. 3 amino acids like threonine, isoleucine, and valine. Propionyl-CoA carboxylase is the only exception where a fatty acid is made to glucose, and the fat acid goes in the TCA cycle through succinyl-CoA, which then enters gluconeogenesis pathway. The last one is Acetyl-CoA carboxylase. Acetyl-CoA, a 2-carbon, is converted to malonyl-CoA, a 3-carbon. This occurs in the cytoplasm, and it is the rate-limiting and the regulated step in fatty acid synthesis. It is allosteric activation by citrate and allosteric inactivation by long-chain fatty acyl-CoA, which is the end product. Also, the last slide here, which I'm just going to make sure that you know, is that it is insulin and citrate activates the acetyl-CoA carboxylase, glucagon and palmitoyl-CoA inactivates. So that's everything. Thank you, and please come again.